Guys, this is a review of biochemistry. We're going to run through this slideshow pretty quickly. We talked about carbon, how carbon has 4 single covalent bonds, therefore it likes to bond with a lot of different things, and so you find it in tons of compounds in nature. It's very, very versatile because of those 4 single covalent bonds. It's those carbon chains like we see inside of sugars and proteins and fats. And basically all organic compounds are based in carbon. When we look at chemical reactions, we have a reactant plus a reactant yields a product. So in terms of this chapter, we could look at a monosaccharide, and a monosaccharide yields a disaccharide. We can have amino acid and an amino acid yielding a protein or a dipeptide, and when water comes out in the process of dehydration synthesis. Activation energy definitely comes into play when we talk about enzymes, and so we'll talk about that a little bit later. So here's the chemical formula, and there's a structural formula. So there's propane and C3H8, and now here it is structurally what it actually looks like. So you should know the difference between a chemical formula and a structural formula. So again, you can watch the dehydration synthesis video that's posted to review, but this diagram is very important because, as you can see, the glucose and the glucose want to come together to form maltose, but the only way to do so is to take a water out. H2O, the OH and the H are highlighted in blue and come off, and the water comes out. And that is the process of dehydration synthesis. Here we see it again with glucose and fructose forming sucrose. At the bottom, the water coming out. To dehydrate means to pull out. Hydrolysis would be the opposite. Hydro means water. To hydrate means to drink a lot of water. It's really great to take into account the formula of that newly formed disaccharide. It is C12H22O11. It is not simply doubling the C6H12O6 because you lose an H2O. It's important. A question you'd see on a test or a quiz would try to fool you and say C12H24O12, and that would be for you to pick, but that's wrong. It is C12H22O11 because you lose 2 H's and 1 O. You can see the water coming into this reaction for hydrolysis. The water with the enzyme sucrase, ACE, if it ends in ACE, it is an enzyme. The water with the enzyme, and then all of a sudden that water reattaches and you split the disaccharide into a monosaccharide and a monosaccharide. So again, it's a reverse. I'm just going to go back a slide. Again, take a look. Here we have 2 smaller molecules coming together for a bigger molecule. You got a glucose and a glucose to a maltose. Now we're going to reverse it, and we're going to go big down to small. Take out water to build, add water to split. And the way I look at it is, the way I remember that is, water will split things apart. If I put sugar into, whether this is a scientific answer or not, if I put hard rock sugar inside of a glass of water or a glass of hot coffee and I stir, it will dissolve. The water breaks down something in there. Okay? So what does water do here? It breaks down the sucrose into glucose and fructose. It's just the way I remember it, that water causes a solid to dissolve. So hydrolysis, in terms of the definition, the breaking of bonds of a polymer to create individual monomers. Okay, poly means many, mono means one. Okay, if you eat potatoes, your body must break down the starch. You need water for that. And if you eat too much, excess glucose is bonded up in glycogen and stored in muscles or liver, and of course that will turn to fat. That's why they say don't have too much sugar, because it'll turn to fat. It'll get stored in your body. Okay, so a macromolecule are giant molecules composed of hundreds of thousands of smaller molecules bonded together. Now, a monomer is a small molecule, so if you link a bunch of them together, it's called a polymer. And if you do a ton of those, you can make a macromolecule. Carbohydrates, lipids, proteins, and nucleic acids are all macromolecules. It's difficult to model them because you need so many of them together. Even if you put a few sugars together, it's not necessarily a macromolecule. A macro means really big, micro means very, very small. Carbohydrates. You hear about people being on low-carb diets. Well, no bread, no pasta, no rice. Well, what are they? Okay, first off, they contain elements CHO, in a ratio of 1:1. So if you think of glucose, C6H12O6, if you reduce C6H12O6, it goes down to 1:2:1 reduction. There it is. Reduce to 6, reduce to 12. 6 goes into itself one time. 6 goes into 12 two times. 6 goes into itself one time. The main source of energy for living things. Plants use them both for energy and structure. Structure because of cellulose. And it's easy to remember cellulose because when you bite into celery, it crunches. And that's what the cellulose is what keeps that structure. Okay, the monomer of a carbohydrate is a monosaccharide. So a single sugar, like this one right here, glucose, okay, glucose, fructose, and galactose, and the formula is C6H12O6. Now, of course, you guys know, we've already mentioned, that the double formula, or the bigger formula of a disaccharide is C12H22O11, because you lose an H2O in the process of dehydration synthesis for the 8 millionth time. I feel like we're saying dehydration synthesis and hydrolysis a ton in this chapter. If you take a look here, we've got sucrose, lactose, and maltose. All three of those are disaccharides. 2 sugars. Remember, saccharide is sugar. Saccharide, sugar. When you hear peptide later on, it's protein. Peptide, protein, saccharide, sugar. Disaccharides, 2 monosaccharides bonded together, like sucrose, is glucose and fructose. Lactose, glucose and galactose. Maltose, glucose and glucose. Now, if you notice, the oses are all sugars. The enzymes for those, sucrase, lactase, and I believe maltase, but definitely lactase and sucrase, are the enzymes that break down the sugar. So what ends in ase, it's the enzyme. So a polysaccharide, poly meaning many, those are repeating monosaccharides bonded together, and animals store excess sugar like us in the polysaccharide glycogen, which will then turn into fat. And plants store it as starch. And of course, you hear people say things like starchy foods, right? Like pasta, bread, rice. And cellulose, we said, is for structure. So polysaccharides, here you go, is glycogen. And you've got branching together of a bunch of glucoses, monosaccharides, to form glycogen. And this is just a diagram of polysaccharides like starch, glycogen, and cellulose, and you can see that they're linked together and they form fibers in cellulose, and they're very, very complex, long links. So let's move into proteins. So we're going to take CHO and we're going to add N, nitrogen, and sometimes sulfur. Okay, so proteins are macromolecules. Now, we're going to talk about the building blocks, which are amino acids. Now, the building blocks of sugars were monosaccharides. Okay? So, now we're going to talk about amino acids being the monomer, or the building blocks of proteins. Okay, the amino acid. So there are 20 different types of amino acids, and if you recall, there are 20 different R groups. And the R group is one of the 3 parts that we need to memorize of a protein. So the major function of proteins, they're hormones or chemical messengers, they're structural with hair, nails, and cartilage, they're receptors that help communicate, enzymes that catalyze chemical reactions, and antibodies that fight diseases. So the structure of an amino acid, you've got the R group, okay, you've got a carboxyl group, and you've got an amino group. Now, the carboxyl group up here the COOH, is easy to remember because it's carbon-oxygen, so carboxyl. The amino group is NH2 on the top, and the R group is pretty much anything else. It changes for different amino acids. There's 20 different R groups, therefore they have 20 different amino acids. Now, this diagram in the lower right here is important because it shows the peptide bond. Now, the peptide bond that occurs after water is removed in the process of dehydration and synthesis is known as a peptide bond, which is why when we bond them together, we call it a dipeptide. We would love to call it a diamino acid, but we don't. We call it a dipeptide. Peptide, protein. Two amino acids form a dipeptide. If you keep linking, you form a protein. Proteins are going to be a macromolecule, a bigger strand. These are a bunch of amino acids. All of them have the 3 groups. So if you take a look at them, I know it's maybe difficult to see, but if you're on full screen, you might be able to see. You should be able to see the R groups, which look different, the COO, and the NH2, okay? And that's the amino group. So you can see an amino group on each one. So if you look in the central one, you see a carboxyl group, and then this bugger down here is the R group. If you look at another one, you'll see carboxyl, amino, and R group. That lonely H and C kind of are the core of every single amino acid. Polypeptide. There you go. Tons of chains together with different R groups. Shape and function. Okay, so a protein shape determines its function. Now, they're all strands, but then they bend in different ways. They kind of crinkle up, and if you take a look at this diagram, it's kind of interesting. So you've got amino acid chain, which is the primary structure, but then what you can see is things kind of bend in different ways, which has them do different things in nature. So enzymes. They speed up chemical reactions and lower the amount of energy needed to do chemical reactions. Most of the enzymes are proteins, and like I said, they reduce the amount of energy. So how does it all work? There's an active site which binds to another molecule, and that molecule that comes in is called a substrate. So the enzyme allows a substrate to come in and do a couple things. Either two small things can come together, or a big thing can come in and get split apart. Combined over and over again, so the enzyme can stay there, and new substrate can come in and leave. Come in and leave. Now, I encourage you to check out the video on enzyme function that's posted. And they're very specific. The binding location looks like almost like a lock and a key model, so only certain enzymes bond with certain substrates. And they are affected by temperature, pH, and concentration. That's important. That can affect the way an enzyme functions, the temperature, the pH, and the concentration. So again, you can see here you have a large substrate coming in. You've got the enzyme substrate complex, and then it's splitting apart. Here you have two smaller substrates in letter B, bonding and forming a bigger one. Again, this just goes over the fact that we lower the activation energy with an enzyme. So you notice the pink lines has a lower activation hump in this graph. It takes more energy on the black graph without an enzyme, less with the red, but the same products. Lipids are fats. Okay, they contain CH and O, and there's really no true monomer. Why do they exist? They store energy, then they form cell membranes, and a couple hormones are made of lipids. But the cell membrane is really important because without a cell membrane, you wouldn't have protection for the cells and the ability for water to pass in and out of them, keeping them balanced in homeostasis. And they're nonpolar, unlike water. So fatty acids, okay, so if you take a look at saturated fat versus unsaturated fat, unsaturated fats have a double bond. Take a look at the double bond, which causes them to bend at a certain point and allows them to stay liquid at lower temperatures. Whereas a stick of butter will stay as a solid, it will be more difficult to melt. Olive oil will be liquid at a regular temperature, like a room temperature. So saturated and unsaturated, difference is the double bond there and about whether they're solid or liquid. They store energy. and there's tons of carbons and hydrogen bonds that store tons of energy. The energy is stored in every one of those little sticks. Every one of those bonds is where the energy is stored. So you can see a solid at room temperature is butter, and then a liquid at room temperature. Now, if you were in the summer and you turn the air conditioning off, the butter would eventually melt, but that's not technically room temperature. It'll be getting hotter. But the liquid oil is already a liquid. It stays there. It's that double bond that causes that bend. If you look at this diagram on the right side, and that bend affects the way it exists in nature and the way it's used by the body, digested, or passed through the system. And triglycerides, tri meaning 3, this is a triglyceride. So the green is the glycerol, and then each fatty acid is the red. So it's how we store fat and transport it. And again, this is the glycerol with the 3 fatty acids. Now, what doesn't change is the glycerol. The glycerol stays the same, but the 3 fatty acids can change. There's a bunch of different types. So phospholipids, that's what makes up a cell membrane. So a cell membrane has a hydrophobic tail and a hydrophilic head, which means the head of it loves water and the tail of it hates water. And that's what forms the inside of a cell membrane. So we don't have to worry too much about these complex lipids, but you have heard of cholesterol and testosterone, but they are more complex lipids and waxes. Now there's 2 types of nucleic acids, and those are DNA and RNA. Both store information in cells. And you've heard of deoxyribonucleic acid, ribonucleic acid, and the monomer of them are nucleotides, and there's 4 different nucleotides. They contain phosphorus in there. Now, the structure of a nucleic acid is important to pay attention to here. You've got a phosphate group connected to a sugar and a nitrogenous organic base. Okay, so what you can see here is the structure forms what's called a double helix. A double helix. Now, if you want to make a copy of this, what you have to do is you have to unzip it just like a zipper, replicate it, and only certain of the bases will go with another one because there's almost a lock and key model. If you notice in the right diagram, the thymine goes with the adenine, the cytosine goes with the guanine with the shape. Okay, adenine cannot go with cytosine, thymine cannot go with guanine. And that is your review of the unit. You should, of course, review your outline and definitely review your lab and your questions from any lab activities we did. I hope this was helpful.